we have a consortium of 19 partner research institutes spread across 11 countries, including experts from across Europe and into Israel, North Africa and Sub-Saharan Africa, to address the outbreaks of blue tongue that have occurred in Central and Northern Europe really since 1998. Prior to that date, blue tongue wasn't present in Europe apart from occasional introductions. But since that date, it's become very established in the south in the late 90s, and then subsequently in 2006 it arrived in Central and Northern Europe and has now become very much endemic in the region, representing a continuing threat to livestock health and productivity. So the PALE-Blu consortium, which PALE-Blu stands for pathogen, livestock and environmental relationships affecting blue tongue virus. We're looking at the way the virus has spread, better diagnostic systems, better vaccines, analysis of outbreaks and how they move, the interrelationships of the virus with the insect vector population and its ruminant host, to try and develop not only a better knowledge base, but also to achieve better control and reduce economic losses.
It is very important to do research and to try to find the blue tongue virus, because this virus induces very important direct and indirect economic losses. Direct losses because the animals are ill or can die, and indirect economic losses because you have to fight against this disease, you have to put in place surveillance, you have to put in place vaccination, and you have also sometimes international trade ban. In 2009 and 2010, there was a compulsory vaccination in France against 2 serotypes of virus with inactivated vaccine, and the cost to the year 2010 was estimated to be 98 million euros. The total cost, for example, of blue tongue serotype 8 for the years 2006 to 2011 were estimated at 200 million euros at the EU level. So I think that these elements show you the importance of the economic losses due to blue tongue virus. And this is why such a project is very important, to develop tools, to develop vaccines, to develop knowledge about the disease.
Blue tongue virus is transmitted by CULICOIDES biting midges, very small insects that need to take a blood meal to complete their biological life cycle. CULICOIDES can become infected by taking a blood meal on a viremic host. The virus infected the midge, and the insect can harbor the virus in its salivary glands all lifelong. Then each insect is able to infect a number of other susceptible hosts during the following blood meals. There are more than 1,000 of CULICOIDES species all over the world, feeding on a large number of vertebrate species. Among these, less than 40, we can say, associated to livestock, are able to transmit the blue tongue virus in different continents. For example, there are some species occurring in the African continent, such as CULICOIDES IMICOLA, for example. Others occurring in Europe, as CULICOIDES OBSOLETUS. And they meet in the Mediterranean basin. The study of the interactions between insects, hosts and virus is a relevant part of the PALE-Blu project.
Next generation sequencing technologies arrived more than a decade ago, and they are changing completely the way we do diagnostics of infectious diseases. This is also valid for vector-borne diseases. Just imagine you have a sample, a mosquito or the blood of an infected animal, and you want to know which are the pathogens potentially in that sample. Next generation sequencing technologies allow us is to identify all the genomes in a sample. So what we just have to do is sequence with these new technologies the sample, and we will have all the genomes, including the potential pathogens in the sample. A single test and we do all pathogens. It looks like the perfect test, and it's probably the case for new emerging pathogens. But the problem is that yet it is rather expensive. It requires sophisticated equipment and high technical skills. But we are working on that. We are trying to develop new protocols to facilitate the uptake of these technologies.
If we can work out what makes the disease move or how it moves, then we can predict where it goes, which means we've got some chance of doing something about it and also warning livestock keepers in the areas to which the disease may move. In order to do that, we have to assemble a lot of different information, different sorts of drivers, we call them, satellite imagery, climate information, things like temperature, rainfall, wind, but also the movement of animals from one place to the other. So it's quite a complex process, and it takes many years. The statisticians can link it to the position of the disease and the outbreaks at different times of year and different years.
Current vaccines has their disadvantages and advantages, so we need a next generation vaccine, and my group is working on that, and we have a very nice vaccine to protect our animals so that the livestock is safe, and we need a lot of livestock for all kinds of productions. So we have several approaches, and we are discussing which is the best one to bring it to the market.
Here in Senegal we have breeds which is a little bit very resistant for the disease, but it can be a source of introduction of the virus from West Africa to North Africa through Mauritania or through other countries. And here in Italy we have already outbreak regarding BLUETONGUE, and we have now a consortium for how to handle and to tackle this problem. Now we have more than 30 serotypes circulating now, and we have also what we call the unknown serotype. We cannot determine them, and it is very important to go further and to sustain this project. It's very important for this connection, because the European Union know that the vectors can just jump from Africa to the Maghreb and from the Maghreb to European, and it's why that now we don't say that I'm safe here in my area.
Prevention could be based on two main actions. First one is early detect the BLUETONGUE virus, and the second is rapid application of control measures. The first one is based mainly on surveillance. The BLUETONGUE virus in Europe has mainly two corridors of entrance. Eastern corridors from Turkey and Middle East, and Western corridors, Northern Africa. So a common strategy could be, first of all, to strengthen surveillance in the border of Europe towards these two corridors. Why it is important to know what is happening in countries bordering Europe? If we know what is circulating in Northern African countries or in Turkey and Middle East, probably we know what will occur in Europe in a couple of years. We have seen that, for example, July 2000, BLUETONGUE serotype 2 was circulating in Algeria and Tunisia, and in August, September 2000, BLUETONGUE virus serotype 2 was in Italy and in Balearic Islands. So focusing our attention on these bordering countries could be a good strategy in order to be prepared to these new possible incursions. The other one is rapid application of control measures. vaccination is the best tool. One of the problems of vaccination today is we don't have sufficient vaccine doses available in the market. Why? Because drug companies prefer not produce these doses because the risk of not having enough order for them is a loss of money. So one possible strategy could be, when we know before what could be the potential circulation of serotypes in Europe, we can anticipate this, preparing a sort of antigen bank. For example, if we know that serotype 3 is circulating in Northern Africa, we can start producing vaccine doses in Europe, so to be prepared of the entrance of this new incursion in Europe.
And the project brings together some of the world's best experts and best organizations for dealing with the virus. So I'm very pleased with the success and the progress we've made.
This video has been realized by the EU Horizon 2020 project, PALE-Blu, Understanding Pathogens, Livestock Environment, Interactions Involving Bluetongue. For further information, visit the website paleblue.eu.